Good morning. My name is Evelyne Jacqz-Aigrain. I am a pediatric pharmacologist working in Hôpital Robert Debré in Paris. We are going to talk today about the implication of pharmacogenetics in drug development. I think it's important to start with definitions. Pharmacogenetics is the study of inter-individual variations in the DNA sequence that are related to drug response, efficacy, and toxicity. While pharmacogenomics has a broader sense, as it's defined as genetic variations, alteration in gene expression, and post-translational modifications. As you see in this slide, the relation between genotype and phenotype depends on genomics corresponding to the analysis of differences in gene sequences related to single nucleotide polymorphism, deletion, or duplication of different genes, but also post-genomics defined in different steps: transcriptomics, studying how genes are transcribed into messenger RNA, proteomics, how genes are expressed as protein, and metabolomics, how genes influence the chemicals that control cellular biochemistry and metabolism. Pharmacogenetics participate to variability in drug pharmacokinetics and pharmacodynamics. As shown here, genetic variants in genes affecting pharmacodynamics will have an impact on the absorption, distribution, metabolism, of excretion of a drug. Genetic variant affecting the pharmacodynamics will have an impact on the target, the mechanism of action, or in drug response. And therefore, the link between the drug and the clinical outcome in terms of efficacy and toxicity will be affected by these genetic variants. The first polymorphisms were described more than 50 years ago. The first one was the polymorphism affecting N-acetylation. And as you can see here, there is a bimodal distribution of isoniazid concentration 6 hours after giving a standard dose to healthy volunteers. And this was related to polymorphism in the L-acetylation of this drug. More recently, in the 1980s, polymorphism of different cytochrome P450 and primarily cytochrome P450-2D6 or the brisequin cytochrome P450 had a major impact on the metabolism of many drugs, including codeine. And what you can see here is that when given codeine, because of polymorphism of this cytochrome, the transformation to morphine is highly variable. And they are either extensive metabolizers who will have concentration of morphine much higher than poor metabolizers when receiving codeine. And this is related to the absence of cytochrome P450 activity in poor metabolizers. The clinical consequences are important because in poor metabolizers, there is no transformation of codeine to morphine, and therefore the analgesic effect of morphine is very low. There are major applications of pharmacogenetics in drug development. First, application in preclinical drug development. Pharmacogenetics is implicated in the target identification of different drugs or tested drug based on genomic data from the Human Genomic Project. In this situation, single gene disease are much easier to study than complex diseases, because in the last case there are many potential targets. The application also includes candidate selection to be transferred to the clinics, taking into account the difficulty of disease heterogeneity, the need to know the mechanism underlying disease, to quantify or variability in drug response, and study the impact on growth and maturation. The implication of pharmacogenetic in pharmacokinetics may be used for retrospective analysis of clinical trial results based on genotype data in order to explore and explain variability in pharmacokinetics, issues in pharmacodynamics, and analysis of adverse drug reaction. And as shown here, there is an important variability in plasma nortriptyline concentration according to the different genotype of tested individuals. In patients that have no functional cytochrome P452D6, plasma nortriptyline concentration are high because metabolism is deficient, while in patients who have normal or even very high activity of cytochrome P452D6, as some of them may have more than 13 genes resulting from duplication. In such case, the concentration of nortriptyline are very low due to extremely rapid metabolism. Pharmacogenetics is also implicated in drug response, and prospective studies may be conducted with selection of patients in different subgroups based on pharmacogenetics to obtain disease subtypes or pharmacokinetic subgroups, either rapid or slow metabolizers, for example. In such cases, we have to remember the potential impact of additional variables on drug response: age, environment, ethnicity, compliance, and diet. According to that, the current status of drug development research consists on studying a population of patients with a given disease and selection patients who will respond to the drug in a subgroup of patients receiving the drugs. The future objectives of pharmacogenetics in drug development research is to subdivide the population of patients with a given disease in patients who will respond to the drug and patients who will need a different drug because they are not responding to the drug because of genetic differences. Application of pharmacogenetic safety is also important. And as shown here, there is a very strong link between the human leukocyte antigen HLA-B star 1502 and the occurrence of Stevens-Johnson syndrome induced by carbamazepine, showing that this pharmacogenetic marker is a high predictor of this disease. As shown here, the link between Stevens-Johnson and carbamazepine was reported in 100% of the patients who were carrier of this specific marker. We need to understand that pharmacology and developmental pharmacology is impacted by growth, maturation, which will change gene expression according to age. Of course, the genotype doesn't change, but the phenotype, as seen here, changes with age. And in many cases for drug metabolizing enzyme, the level is quite low at birth and increases progressively but rapidly after birth to reach almost adult values between the age of 5 to 15, depending on the metabolic activity tested. And this is obvious that we have to take into account the impact of growth and maturation on gene expression. And this will explain why the importance of genetic variability is different between children and adults. As conclusion, there are several potential goals for pharmacogenetic in drug development. First, to ensure the subdivision of common diseases into different molecular subtypes, which may be more or less susceptible to specific treatment. To involve more logical approaches to dosage efficacy and the prevention of adverse reactions by analyzing the genetic basis for differences in the pharmacokinetic or pharmacodynamic properties of drugs. To identify genetic susceptibility to various common diseases that offer targets to pharmacological intervention. There are multiple perspectives in the omics science, including genomics, transcriptomics, proteomics, and metabolomics, to link gene and genotype to phenotype. And all these new sciences will help to understand the biology and the pathology of different diseases. There are multiple environmental factors that will impact the expression of genotype to phenotype and modify metabolomics. This includes genetics and epigenetic factors, age, environment, diseases, drugs, nutrition, and lifestyles. In conclusion, there are multiple perspectives carried by bioinformatics to integrate all the informations that will lead to integrative pharmacology and integrative medicine. First, it will be important to integrate the information issued for lifetime exposures. And here are listed some of the major factors that may impact drug disposition and effect, including, for example, pesticide, medications, tobacco, physiological stress, that will be analyzed to understand their impact on pharmacokinetics and pharmacodynamics through different steps: analysis of the epigenome, of the transcriptome, the proteome, and the metabolome, to understand the relation between dose and response, and then define an health phenotype that will be used to select the most adapted treatment. Thank you for your attention.